All right, so we're going to take a look here at DNA replication. So first off, I want you to actually pull up your, or pull out your written notes. And this is going to be the last little bit that we actually put into there. So I want to kind of take a minute to kind of keep this in mind as we continue on here. So we have these four words here: nucleus, chromosome, gene, and DNA. And we use them an awful lot in regards to talking about genetics. And I think it's really important that you understand how these relate in terms of their size. So we're going to start over here on the left-hand side with the nucleus, and that is actually the largest component. So when we talk about the nucleus, we're talking about everything. In other words, it contains all of the subunits of chromosomes, genes, and DNA. If we take it down to the chromosome level, there are multiple chromosomes in a nucleus, and that is a specific discrete unit of DNA that's been coiled up. If I look on a chromosome, a specific region of that area is called a gene, and that gene, all of the genes are made up of DNA. So DNA is kind of the smallest component of this in terms of size, all right? So when we're talking about DNA, and we need to, we've talked about that double helix, we've talked about how the base pairing rule works, and so now we need to figure out and kind of relate this back to mitosis. We talked about the replication of DNA during this process. And remember, the replication of DNA is the process of copying DNA in the cell. And keep in mind why this is important, and that's because we have to have our daughter cells from mitosis be genetically identical to the cell that it started from, the parent cell. And so we have a quick little reminder here of what's going on during mitosis. So as you can see in here, we start with a black chromosome and a white chromosome, or not a white, or red, and they're kind of built like that ladder to represent the DNA. And you can see we kind of pull it apart, and then all of a sudden we have two of them. So we're kind of focusing on this stage here, how we go from having one of each to now having two of each. So now that we understand the structure of DNA, we can actually look at how it replicates, because the structure is what uniquely allows it to replicate. Each of the strands, so each side of the DNA, has all of the information that we need in order to construct the other half. And we kind of practice that with the base pairing rule. If I only gave you one side, you knew automatically what the other side was. So essentially all we have to do is separate those strands and follow the base pairing rules, and we will actually make new strands of DNA. And that's pretty much what happens. So if I have one side that has a code of ATG, CTT, GGA, TCAA, I want you to actually pause the video, and after you write that down, which I know takes a little bit, I want you to actually write what the other side would be based on the base pairing rule. So go ahead and pause, and then you can come back and you can check your answers. Okay, so how did you do? So because of this, we automatically know what the other side is going to be. And so since I have two sides of the DNA that I am pulling apart, I can actually copy both sides at the same time. And because I can copy both sides at the same time, so I pull apart the strands, I match up on one side to create a new strand, I match on the other side to create a new strand. This is why we call replication a semi-conservative model. And that is because the new DNA molecules that are made are a combination of the one old strand of DNA, so to speak, and a new strand, a newly created strand of DNA. So that double helix is a combination of new and old DNA. And you can kind of see this here in this diagram, which is a little bit easier to see in color than it is in black and white, which is why I like it. So the old strands here, you've got the old blue and the old red, and you can see here it separates. And so now I have a new red and a new blue that is forming. So now I'm going to have two new strands of DNA formed from the single old strand of DNA. All I have to do is separate and start matching up those bases. So the first step in DNA is that we actually have enzymes that come in and it unzips the molecule. And what it does is it breaks the hydrogen bonds between the strands. So we actually pull it apart there right at the hydrogen bonds. Then we have a specific enzyme come in called DNA polymerase. It brings in the new nucleotides and joins them together. So it kind of glues them together or sews them together. So, like I said, it uses that original strand as a template. So the copying happens in opposite directions, so along the two strands and in multiple places at once. So for one strand, this is called the replication fork, okay, where it splits. It's just going to keep adding new ones here. This one here is going to go this way, and on the one side, it'll just be continuous. On the other side, we're going to get little fragments that have to get stitched together. And we'll actually see that in a little bit of a video clip that we look at next. But you can kind of see how this happens. So we're untwisting here, and we're starting to create new strands of DNA. And we're going both to the left and to the right to create this. So how accurate is this process? I mean, we have 3 billion base pairs. How good of a process is this? And I mean, for some of our cells, this is happening all the time, like our skin cells. We try to keep DNA, there's a proofreading process that takes place, and it keeps the actual accuracy pretty high and the number of problems pretty low. So the error rate is down to about one mistake, one error every 1 billion nucleotides. So if you think we have 3 billion nucleotides, so we get about 3 mistakes every time we copy our DNA to make a new cell, which actually isn't bad. But there are things that we are exposed to that can actually increase that. And that's what a mutation is. Those are things like chemicals that we're exposed to, as well as radiation. All right, so before we kind of move on with replication, I'm actually going to have you guys, there's a video here. I have the link on Schoology, and you guys are going to go ahead and you will watch that video.